Okay, our next speaker is Eric Rubin from Merck & Co., and he's going to talk about novel strategies for drug development in cancer and an industry perspective. Eric, you're very welcome.
So first I'd like to thank Dr. Chabner for inviting me to talk about Merck's strategy in oncology drug development, especially for following my former mentor, Dr. Livingston. So you may have seen versions of this slide before, which is a scorecard for oncology relative to other therapeutic areas for about the past 50 years. So you can see that in contrast to heart diseases, cerebrovascular diseases, and infections, we really haven't done too well in oncology in terms of decreasing mortality rates. And it's well known that oncology drugs have a higher failure rate in phase 3 than other therapeutic areas. About half fail due to lack of efficacy, a quarter due to failure to improve on available therapies. And so we really need higher bars and better drugs. In addition, for drugs that are approved, such as this one, that's now a standard of care for this particular disease, you could argue that although these survival curves, the difference between the previous drug and the current drug, the previous drug is the gold and the new drug is the blue, that although statistically significant, is really not a huge win for patients. So Merck's approach to this, similar to others, is to try to find the right drug for the right patient. And this slide shows the preclinical efforts we're using for that. So we like to use a knowledge-based target selection. We're using synthetic lethal screening quite a bit, and I'll take you through an example of that. And we also use next-generation molecular profiling to identify our best targets and then use chemistry and pharmacology to advance those to the clinic. On the clinical side, we spend a lot of time on target engagement assays to make sure that we're hitting the target before we proceed. We like in phase 2 to have what we call a killer experiment, which is it may not be a large population, but a population that should respond to the drug, and if it does not, then we'll kill the drug. This, of course, requires a functional test that's CLIA certified. And then finally, in phase 3, our approach is to find subpopulations in phase 3 that will benefit from the drug. And again, this requires co-development of a diagnostic test. So to do this, we've invested in collaborations that may be of interest to you. One is a collaboration with Moffitt, where Moffitt provides biopsies from basically all patients who enter their doors. They're over 5 years to provide about 25,000 samples. We're now, I think, you can see the figure over here, we're in the several thousands at this point with a variety of cancer types that encompass both primary and metastatic tissues. So we obtain and share mRNA profiles from all these specimens. In addition to mRNA profiling, we're also exploring whole genome and exome sequencing, as well as genome-wide methylation analyses. We've also expanded this to Asia in collaboration with Lilly and Pfizer, with the idea is to collect several thousand lung and gastric tumors samples, as well as associated normal tissue, and to build a similar database of molecular as well as clinical data that will allow correlations and identification of new pathways, as well as responsive subpopulations. So I wanted to give you one example of how this is translated into something that we're now using in the clinic. And this is a RAS gene signature. Again, you're familiar with the importance of the RAS pathway in cancer. We took the approach that having a signature would be useful in identifying KRAS mutant tumors, as well as wild-type tumors that had a RAS dependency phenotype. So this slide shows that we were able to do this. This is really based upon Moffitt data. This is an example of a lung tumor panel where the red bars indicate tissues that contain mutations. The blue is the RAS signature score, and you can see that many of the non-mutant patients have a high RAS signature score, similar to that of the mutant patients, and that if we define a patients as having the high signature score as being RAS dependent, we approximately double the population of tumors that we would think would be susceptible to a RAS targeting therapeutic. We've also shown that the signature correlates with sensitivity to, as expected, to drugs that target AKT, our AKT inhibitor, MK2206, where this high RAS score confers resistance, whereas in contrast with a MEK inhibitor, we see the opposite phenotype. So one of the issues in drug development that I think you'll agree with is that the most effective treatments require combinations. Really, most of our curative therapies involve drug combinations. And these are the principles that really have not changed over many years. The idea is to have additive or synergistic anti-tumor activity without additive toxicity. However, accomplishing this in practice is actually fairly difficult. And we've been using two approaches that we hope will result in more rapid advances in this area. One is, as I mentioned before, using synthetic lethal approaches preclinically. And the second that I'll talk about are new clinical designs, new clinical trial designs that hopefully will get to the most active combinations more quickly. So this can be called a combinations problem. And the traditional clinical approach of multiple phase 2 trials with exploratory or retrospective biomarker objectives really doesn't work. There are too many such proof of concept trials to prosecute cost effectively. So for example, if you have 10 new drugs in a field, that gives you about 45 concurrent trials, and that's without biomarker enrichment strategies. In addition, targeted drugs are unlikely to be effective for all patients in a given cancer type. So again, there's a multiplier there that you need to be concerned about in terms of finding the predictive subgroups. And I'll give you an example of that a little later in the talk. In addition, the traditional development pathway of determining the maximum tolerated dose, followed by a CT-guided efficacy study and then a pivotal phase trial, is inefficient and really hasn't worked very well. So the remainder of the talk, I'm going to give you a couple of examples of how we've tried to do a little better. One is we try to identify promising combinations in a little more agnostic fashion preclinically. I think it's very easy to put two drugs together and come up with an argument that they should be a good match. And indeed, there's a host of preclinical data that where one might show that these are additive or even synergistic. But in many times, when those combinations get to the clinic, we find they're really not effective. So we have two that we are particularly interested in. One involves a combination of an IGF-1R targeting compound with an mTOR targeting compound, the other with an AKT inhibitor and a MEK inhibitor. And again, the second part is, as I mentioned before, is to use new approaches to combination trials that are adaptive in nature. And examples of that are Battle2, which I'm going to talk about, and as well as I-SPY2, which I'm not going to talk about. So with regard to the preclinical approach, as I mentioned, we do use enhancer screens using siRNA or shRNA libraries, as depicted here. These give us candidate enhancers, which we then validate. So a good example of that is this figure, which shows a screen for enhancers for MK0646, which is a monoclonal antibody to IGF-1R. So what these dots are individual shRNA results, where the x-axis is proliferation. So dots that are more towards the left, there's an anti-proliferative effect of the shRNA. And the y-axis is basically the p-value, with higher being more significant. So you can see there's lots of dots here with a real outlier up here, which is the mTOR gene, FRAP1. So again, this, in an agnostic fashion, really identifies this particular combination as being something special. And happily, the converse was also true. So if we switched it and screened for enhancers of the mTOR inhibitor, Rodafrolimus, we identified IGF-1R as a top hit. So this provided a rationale for the combination. Happily, this made sense biologically. It had been shown, some of this work done by Dr. Baselga, that there's a feedback loop that if one inhibits cells with an mTOR inhibitor, you activate the AKT pathway. And so by blocking that, with an IGF-1R inhibitor, for example, one could stop this feedback loop. In addition, in preclinical animal models, our data showed that this combination indeed was synergistic. So this is one slide of a patient on our phase 1 clinical trial of this combination. Patient with high proliferation breast cancer, ER positive, PR positive, HER2 negative, and high Ki67. This patient had 4 prior chemotherapy regimens, 3 prior endocrine regimens, and had a quite dramatic response to this combination with a confirmed and sustained partial response, improved pain, and as well as a decrease in a tumor marker. And indeed, 4 out of 7 patients with this phenotype, this high proliferation luminal B type phenotype, have had objective evidence of response. So quite striking response rate in our phase 1 trial. And we're pursuing that. We've actually just started a phase 2 trial in that population with that combination. So we also have an approach that, again, we hope is an agnostic way will help us define these types of combinations that involves what we call the OncoPolyPharmacology, or OPP. That's a robotics-based system that involves hundreds of cell lines with 2 different drugs and a matrix of interactions where you get up into the millions of combinations. So this is, and I'll show you one example of this output from this on this slide, where there's a measurement. This actually should be HSA of synergy, and again a p-value, and down here it just shows this synergy parameter is based upon a 2D surface type assessment of drug interaction possibilities. And so using this particular model, we identified a particular combination as being highly active across multiple cell lines, and this was a combination with AKT inhibitor and a MEK inhibitor. So this is also supported biologically by understanding the pathway, as well as by cellular experiments that indicate that pathway flux is inhibited to a much greater extent by the combination than by either single drug alone. And this, in part, led to our collaboration with AstraZeneca in combining our AKT inhibitor with their MEK inhibitor. This was one of the first such collaborations in an early stage. And we're in the midst of the phase 1 trial with this combination currently. So I'd like to talk now about the clinical approach that I mentioned before. This is, again, back to what we would call the combinations problem. This is an example in lung cancer, a simplified example, where if you take 2 standard of care drugs, in this case, let's say, erlotinib and bevacizumab, and let's say you're a company that has 5 new compounds that you're interested in lung cancer listed here, and maybe you would say that matching those compounds might be 4 predictive biomarkers that you'd be interested in. So if you just do the multiplication, you get to 20 possible all-comer phase 2 trials. If you look at the biomarker groups, assuming they're independent, which may not be true, but for simplicity, we'll assume that's the case, you get to 16 possible subgroups. So if you do the multiplication, you're up to 320 possible enrichment phase 2 trials, which of course is not feasible. So how can we, what's the approach here? Well, one that we became very intrigued with was the approach taken by the lung cancer group at MD Anderson, and really based upon adaptive statistics from Jack Lee and Don Berry. So this is the BATTLE trial, which you may have heard about. It was presented at AACR plenary this year. So BATTLE stands for Biomarker-based Approaches of Targeted Therapy for Lung Cancer Elimination. This is really an umbrella trial that has 4 sort of separate phase 2s in a second-line non-small cell setting. So the BATTLE group, they were interested in 4 biomarker groups and realized that, again, they had that results could be positive or negative for each of these 4, giving you 16 possible groups. If you multiply 16 times 4, again, you get 64 combinations. That's too many phase 2s. So what they did is they used a clustering approach and separated the biomarker groups into 5 hierarchical groups, which yields only 20 possible combinations, which was feasible for them in a single institution study. Importantly, they used adaptive randomization to identify the most active treatment. So this means that as patients enter the trial, their probability of being assigned to one of the arms according to their biomarker group changes as a function of how other patients have done in the past. And statistically, with only 200 patients, although this is a Bayesian adaptive, it's not a frequentist type trial, it yields type 1 and type 2 error that are not dissimilar for typical types of phase 2s that would be more traditional. So this is, again, the schematic for the trial. So all patients entering the trial had a biopsy, and they were randomized to initially equally and then to this adaptive design that I mentioned. Here are the 4 different treatments: erlotinib, VEGF inhibitor, vandetanib, erlotinib plus bixaritine, an RXR inhibitor, or sorafenib. The primary endpoint was an 8-week disease control rate. So I don't have time to take you through all the results, but I'll mention some of them quickly. So here again are the 4 biomarker groups that I mentioned. There's also a 5th, which was basically patients not having that group. And so they were an EGFR marker group, a KRAS or BRAF group, a VEGF group, and an RXR or cyclin D group. These were roughly designed to match to the arms that were being studied. Here's data that argue that the 8-week disease control rate is actually relevant as an endpoint. You can see that it does correlate with survival, and patients who, with a p-value of 0.02, patients with disease control at 8 weeks had a greater median survival relative to those who did not. And here's the results according to arm and biomarker group. So first I'll draw your attention to the overall response rate, if you will. It was 46%. So again, that's pretty good for a second-line lung cancer population, and argues that this is actually good for the patient. So patients are, one could argue, are being placed onto trials that match their characteristics more closely. If you look in sort of some of the particular cells, you'll see that for erlotinib, not surprisingly, patients who have EGFR mutation have a fairly high response rate, in this case 35%. One of the interesting ones was sorafenib, which had been studied in all-comer lung studies and had been negative, was identified as having a very high response rate in patients with KRAS or BRAF mutation, 79%. And indeed, I think that stimulated some additional work on sorafenib as a treatment for those patients. So we're working with MD Anderson on what we call Battle 2, which is a next iteration of this idea using combinations that we're interested in. And these are a combination with our AKT inhibitor with erlotinib, as well as the MEK AKT combination that I mentioned previously. It's the same type idea where patients will be stratified according to biomarker groups. Initially, this will just be EGFR mutation and KRAS. But we have an interim analysis where part of the trial, the beginning part of the trial, will actually be looking to identify additional predictive biomarkers. We call these Group 2 biomarkers. These include a variety of pre-specified biomarkers that have been picked in collaboration with MD Anderson and us, as well as with their investigators as part of their program project grant. So I'll just comment, though, that one of the other issues with combinations like the IGF-1R mTOR or the AKT and MEK is that there's a challenge in getting 2 new drugs approved at the same time. Again, you might agree with me that that if we're really going to accelerate drug development in cancer patients, this is important. There's currently no precedent, and FDA guidance and regulations have lagged. There is something called the combinations rule at the FDA, which is really based upon combining 2 approved products in one pill. Merck has actually done this very well in other franchises where one can combine an anti-lipid drug with an antihypertensive drug in one pill. That's more convenient for the patient. But that's not really the same idea for cancer. And these requirements really, you have to, for approval, show that there is a unique effect of the combination relative to either monotherapy. If we follow this in oncology, we would actually have to enroll patients onto typically a 4-arm phase 3 pivotal trial where patients would be exposed to treatments, monotherapy treatments that would likely be ineffective. So happily, the FDA has recognized this and is coming out with guidance very shortly that will allow both in oncology and in other therapeutic areas more rapid development of, and the possibility of having 2 new drugs approved at the same time. So finally, I want to end with just one other thing that we're doing that we believe is important in rapid drug development in oncology, and that's a collaborative trial network that's worldwide. See, the idea here is that we want to improve the efficiency of pre-proof of concept studies, and we're interested in sites like the one here that are interested in new designs, complex biomarkers, and adaptability. We hope to maximize efficiencies through long-term relationships with these sites, and that things that can take a long time, such as administration, contracting, legal aspects, will be minimized. So we have sites, now we have 17 sites that we're working with worldwide that include, in addition to sites here, sites in Europe as well as Asia Pacific. These are shown schematically here. And so I'd just like to end thanking people from Merck who have helped with this strategy, including Gary Gilliland, Steve Falwell, Elliot Barr, Kevin Anderson, our statistician, and others, as well as collaborators from AstraZeneca who are listed here that have been important with the MEK inhibition, Dr. Baselga, who's been very important in the IGF-1R mTOR combination, and colleagues from MD Anderson, including Roy Herbst, Vale Papamadakpoulou, Jack Lee, and Don Barry, who've been important in the battle design. So thank you very much for your attention. I'd be happy to answer questions.
Okay, questions?
I was wondering if you could say a few words about the REST signature you presented, how you checked it for specificity, that it's really REST specific, it's not proliferation or PI3 kinase specific. The reason I'm asking is that, yes, there was REST signature published in the literature, which was based on primary cells in transient transfection. And for the past 5 years, with Tan Inche from Bob Weinberg's lab, we have produced several stable, transformed cell lines, and we couldn't find anything specific of REST signature that could be, applied to patient population.
So we do have, actually, we do have PI3 kinase and proliferation signatures. And so although there is some overlap, the RAS signature is about 100 genes or so, it is different. And one of the ways we got this, though, was from primary tissues. Again, it goes back to the Moffitt situation. So this is where we looked across multiple cancer types, again, looking at those cancers that had RAS mutations that across different histologies to get the signature. So,
Bruce.
Eric, one of the primary things in the battle trial is to try to integrate VEGF inhibitors or pathway inhibitors with other target inhibitors. The problem I see with this is that is there really any understanding of biomarkers for VEGF response?
Yeah, probably others in the audience can answer that better than me. I'm not sure. I mean, but so one of the issues is you're right. I mean, if you had to sort of bet on picking a biomarker ahead of time, you probably would have a tough time doing that. One of the advantages, though, of this type of approach is that there can be a discovery component. That's what we're doing for the other one, where you can use profiling or something else, a more agnostic way of doing this. And just one comment I'll say about, because I was on a panel with Richard Simon a couple weeks ago, where he's also published in the idea of arguing that we should be a bit more open to both identifying and validating predictive biomarkers in later stage trials. So he's published an idea that for a pivotal phase 3, one could actually start the trial not really knowing the right biomarker. At some point through the trial, stop, identify that potentially predictive biomarker, then the rest of the trial, validate it. At the end, one would have both an approved drug and a biomarker. Interestingly, the FDA at this meeting seemed to be okay with this. I mean, we'll see in practice whether they would allow such a thing. But I think it does get around this bind of and that's one of the arguments even for Battle 1, is that some of the original biomarkers that were picked really, in retrospect, probably weren't the best ones. Yep.
Just in relationship to the issues that you raised about giving 2 drugs and having
Yeah, so again, MACE, maybe we talked about that yesterday, I saw MACE earlier. I think that this is a big issue, and part of it, again, is that it's not to have something that really works in the clinic is not trivial. Translating the basic assays to the clinical assays is very difficult and costly. So again, from a development perspective, you have to decide, when do I make that investment? Particularly, you really have to do it oftentimes before you really know if your drug's going to work or not. Yeah.
A question here that I think that adaptive design is really a great idea to speed up the process. It's here.
Oh, hi.
But I present a counter argument that is, if you have a side effects, you have a combination of two or more, so which drug you're going to blame or you find the cause of it? Or it's a combination of the two maybe causing this unique clinical side effects. How do you deal with that? You may have to go back to monotherapy somehow.
So it's a good question. At one level, I would just say that I think it's very difficult to attribute side effects to one drug. You can argue, even if let's say you're adding on to a drug that has no neuropathy, if that's occurring, you could argue the second drug is actually making that pre-existing side effect worse. But what you're touching upon, though, is really the issue of sort of dose for the two drugs. And so it is an important area where when you combine two drugs, you may find that they're synergistic not only against the tumor but against the normal tissue, right? So that's an important part that gets back to having the right target engagement assays to make sure that in the early stages you're actually making sure it's a viable combination and that you're, in terms of finding the doses, you're finding the best possible doses between the two compounds. And again, we and others are using adaptive trials for that as well.
Any other questions? Okay, well, Eric, thank you very much for a wonderful talk.